Hello, welcome everyone here and everyone online. Welcome to the fourth day of the symposium. My name is Ling Lam. I'm a faculty member at School of Creative Media, CityU, University of Hong Kong. So today we are going to have this plenary fourth section in science and creativity. So I think this is a very interesting and important session because this conference is all about creativity, art, and computation, and of course, machine intelligence. And it is also important to get to know one of the important stakeholders, which are the scientists, to share their view on the topic. Without the overdue, before I introduce our plenary session speakers today, I'm going to state this disclaimer. This session is being recorded and will be available to register at Art Machines 2 attendees until 15 September 2021 at the conference website. It will not be accessible to the public. For online participants, if you do not wish to be recorded, please exit the session and turn off your camera. And the next speaker is next to me, Dr. Rosa Chan. Actually, I have known Rosa for 20 years. Yes, because we went to the same college and we also attended the same program. We also went to LA for our research before we joined CityU as faculty member. Sorry to disclose our age. Okay, so Rosa is currently an associate professor in the Department of Electrical Engineering at CityU, University of Hong Kong. Her research interests include computational neuroscience, neuroposthesis, and brain-computer interface applications. She was the co-recipient of the Outstanding Paper Award of the IEEE Transactions on Neural Systems and Rehabilitation Engineering in 2013 for their research breakthrough in mathematical modeling of the hippocampus for the development of cognitive prosthesis. Dr. Chan was the chair of the Hong Kong-Macau Joint Chapter of IEEE Engineering in Medicine and Biology Society in 2014. She is elected to the IEEE EMBS Administrative Committee as Asia Pacific representative, and she is currently an editorial board member of the Journal of Neuroengineering. The title of her talk today is Eavesdropping on Human Behaviors, Lessons from Engineers. So Rosa, over to you.
Thank you. And thank you very much for the invitation to speak here today. It's very exciting to talk to a very different crowd. And thank you very much for the kind introduction. So I'm from engineering, and so what I do is mostly on modeling of human behavior and mostly modeling of how the brain works. So, and I asked a friend, so when I'm drafting the abstract for this talk, so I write in very technical terms. I did not change my abstract finally, but I asked for my friends' opinion anyway. So what can I do to make it more appealing to artists? So that's my friend's suggestions. So words adding hacking into the human mind, cybernetics, feedback loops between users and designers, material body mind, sino something, post-human something. So so in short, I would treat this as my table of content of what I will talk about today. So what I try to do to show you is that what is a trend in terms of how to build models, what allows us to build better models, and what are the limitations. when we are trying to study the human brain. And I can even further summarize what I do with this. That is, in short, I try to find a relationship between inputs and outputs. And so for here, let's say I want to understand like, how human brain or how biological neural circuits works. What I have here is I have inputs ranging from the environmental observations, audiovisual stimulus that we present to the subjects, or electrophysiological recordings from other parts of the body. And the outputs can be like motion, emotion, or electrophysiological recordings like from other that we like collect from any parts of the body. So this can sort of summarize what a lab does. And when we are trying to interpret like the brain or as a sort of information processing system, so for engineers, we have like many, many years of experience. So usually we always quote like David Marr, which is one of the pioneers in computational neuroscience, and he is expert in vision. So there are 3 levels of analysis for information processing systems. The first level is a computation theory. So we try to understand what is the goal of the computation, why is it appropriate, and what is the logic of the strategy by it can be carried out. And then the second level is that we try to understand the representation and the algorithm. So how can it be modeled, and what's the relationship between inputs and outputs, and what is the algorithm for this transformation. And the third level is that after I have this like mathematical representation, can I implement it and replicate it on the hardware? So like most engineers, when trying to analyze any systems, we follow sort of these 3 different levels. But based on like what we have been doing in terms of modeling of the brain, I have to say, like actually, it's easier for us to replicate the outputs based on the inputs we present to somebody, and we can even like replicate in such a way that like that can be implemented as on a hardware, but understanding what exactly inside and what's the logic may be more difficult than we think. But overall, like throughout all these years, you can see like you listen to David's like a really nice talk, and you may have also aware of a lot of nice work like in this conference. And we have a better understanding of human behavior, ranging from research or like engineering or different fields. We have a better understanding because I feel like there are 3 things have driven us. First of all, is the advancement in neuroscience. We understand the science better. We understand the psychology better. And second is the tools. That is like we have way more powerful hardware. And the third thing is that we have built more better models like through smarter algorithms. And if we talk about behavior in this talk, I will focus on those like motion-related behavior. So when I move my hand, when I'm speaking, actually my brain is sending electrical signals to different muscle groups. And when these muscle groups contract, then I generate different gestures. And inside my brain, there are like 86 billion neurons, all right? And then these neurons have very complex structure, and they vary across like brain areas and different functions, like with like very different structures with different functions. But for engineers, how do we understand? As we mentioned, we need to do some measurements, right, to understand the system. So the first thing is that we got to know what kind of inputs, outputs can we get from the system. So if we just take a look at the brain, so there are 2 more easier ways that we can get the information. First way is the electrical activities, because like when a neuron activates, its voltage changes, and where a lot of neurons activate, then it has a very distinct pattern of electrical activities. And the other way is chemical activity. So in short, when a neuron activates, it needs energy. So that leads to changes in the consumption of sugars and oxygen. So in some sense, the matrix was right, partially. All right. So what Morpheus said, yeah, is like with the help of electrical activities, actually we can interpret what's going on in the brain in some sense. But of course, there are different like recording methods when we are tapping into this like like these activities. So one way is that if you want to do the scientific part, if you want to have more accurate measures for scientific research, what they do is that they have to do surgery, let's say on rats, or experimental animals, and then you put electrodes inside to measure in the individual neurons' activities. That is so-called the invasive recording methods, and you just put probes that measure the electrical activities. And the other way is that is a non-invasive recording methods. So we can put electrodes on the surface of the scalp, and you measure something less, with a much lower resolution, spatially and temporally. So that is what you call like EEG or brain waves. So when we are sitting in the brain, we can do these measures, and then not only central nervous system, when we think about the peripheral nervous system. So let's say I move my hands, then so there's electrical signals going to muscle activities, and these muscle activities can be also measured in electrical form too, and also kinematics, I have motion. So there are various ways we can address all this motion. And my team, even we found that we can, by putting microphones at different parts of your of your hand, we can even tell the hand gesture. So there are different ways to measure the activity. So the next question, before we finally go back to how we bridge brain-machine interface together, is what kind of advances in terms of the hardware and software tools to study the biological neural system. Everything from engineering that goes back to Moore's law in some sense. In short, we want to do things faster, and there are tons of engineers just to make things calculations much faster. And you heard about Moore's law, and we know that our computational power increases through time, and then even the energy consumption theoretically to calculate the same thing it costs less throughout the years. What does it lead to? So higher computational power, and along the way you'll find that there are huge advancement in the wireless communication, and also you can see a lot of more capable sensors, and ultimately you have a lot of low-cost, real-time big data from everybody. So what can you do with it? So you have different forms, ranging from text, audio, images, video, sensor data ranging from motion data. That means even I walk to somewhere, every steps I moved, or electrophysiological recording, you can measure now EKG, related to your heart activities, or your EMG. You can have tons of data from humans. And why I also want to emphasize about tests like audio, etc., because ultimately, when you press a button, when you decide certain things, all these are related to motion, right? And also, when do you think images or video is interesting? You press certain selection. Every kind of this data relate to motion. And so from here, from all these behavior, we can sort of tell, that expressed from our brain, we can sort of try to map from this data and relate with what exactly the brain is doing inside with some of the sensor data too. And from there, with all these data, electrophysiological recordings, now we can start to build mathematical models that bridge different parts of the body. Let's say we separate into three major roles. First is the sensory function. Let's say I hear something, I see something, that is I taste something, those are sensory function. And then the second part is the cognitive function. I process all these data, and then I decide to do something. I decide to move my hand, I shake my head. And then the third part is obvious, the action. After I send this instruction to my hand, to my different muscle set, I decide to do something. So these are actuators. So for the sensors part, after we build this kind of mathematical models, let's say from sensory, let's say image, how does this stimulate the retina? So we actually have now in the biomedical engineering field, you have artificial retina that is actually implanted and restore some of the eyesight. What you see here is this little picture over here. It's a real like artificial retina setup. So you have like a wireless like transfer of like signals and power to some implants in the eye, and the camera is right over here. And a more popular like neural like implant is like a cochlear implant over here. So, and that helps a lot of people to restore the hearing function. And you may also already seen that, let's say for somebody who unfortunately like lost their limbs because of some accident, you find there are lots of initiatives that can bridge like the surface muscle activities, those electrical activities that is measured at the stump, to control certain robotic arm or limbs to help them to regain some of the original motion function. And back when I was in the US, I also participate in a team that does like deep brain stimulation. So particularly for those who cannot encode short-term memory into long-term memory because of Alzheimer or like trauma, and try to restore some of the lost like memory encoding function. And all these like now like getting more popular now. So all this research have been there for like ongoing for decades. It becomes very popular because now the industry becomes like interested too. For example, like Neuralink, like last year they showed a very nice setup that can stimulate over 1,000 channels, that can measure over 1,000 channels, and then from implant. So the cool things about this Neuralink device is that it's very sleek, and the surgery time is much less than what you do in a research lab. So usually if you have a single research lab, it's very difficult to get this kind of like device out. So it's good to have some industry interest to make it happen, to make a better recording device. So far, I don't know about the long-term like stability of these like measurements, but and they already moved to monkey experiments, and they show, like you may have seen like in April, that this monkey using this like implanted device, it can like wirelessly control two cursors playing mime pong, so which I found pretty cool. So, but overall, the key idea is that when you bridge from the mind, the electrical signals you measure from the brain to something, so you need a mathematical model to do this like transformation. So how do we do modeling? So at the beginning, people were thinking about, okay, how about I model from every single neurons and then to networks. So what you show here is just like a very small part, like in the simulation of a part of a neuron. It's not a complete neuron. So already it will cost a lot of time to do simulation for one neuron. If you do it mechanistically, that means you want to study every single, simulate very accurately for every single neuron. It will take a lot of time. So just like the IBM Blue Brain project, so which you may heard of before, and now is lost a lot of like, it does not have much news these days, but the fact is that because it's a very challenging problem, all right? So to build a mechanistic model of like every single neuron, all the synapses, how they work together. And you cannot wait to do a simulation. Let's say if the monkey's mind is thinking something, it will give an output like 1 week later, right? So this is not something that is like possible. So instead, like we are going back to what we talk about for engineers. We do a lot of input-output driven models. That means, in short, like we have a sort of a lower resolution, but like we get this like input neural signals data, and then we try to map based on some experiments, like we try to map it with the behavior, like, and we build this kind of model. And the neural signals that we can use ranging from spike trains, from like brain waves, or there are also other means that people are exploring. So when I was in US, like when we talk about mathematical models, so actually we take a lot of steps to address the dynamics, to address the nonlinearity, to make it more efficient by reducing number of coefficients, to address the fact that it is stochastic. That means in different observation trials, it can be different, even if it's the same system. And it can be even time varying, you know? So the brain is very sophisticated when we try to build a mathematical model. But successfully, like we did this kind of closed-loop simulation on a rat, and back then we showed that it can restore some of the lost memory encoding function, like even like we destroy part of the original biological neural circuit, and we bypass it with this kind of like stimulation. And after I left the group for a number of years, like as you may heard of, like for some patients with very severe seizure, so they have to like do some surgery to locate which region to brain region to remove, so that their seizure can be controlled. So for these like patients, they had like they go through also an experiment that you have stimulation. So that's why like the task at that time in parallel that they do is that they have to do a sort of a human memory encoding and recall task and show that, okay, this kind of deep brain stimulation strategy can actually facilitate some of the memory function. But I left the group for a couple of years already, so I don't know what's the status of that part of research. But what I try to say today, and I hope that another key point is that like you may heard of a lot of like different promising like aspects of like building mathematical models, machine learning. And like today, as engineers, like usually the engineers, we all have to tell you some bad news too, you know. So and what kind of things you have to be aware of when you're building this kind of like mathematical models. So first question for you, okay? So now let's say you take a look at this like this block box called F. You give it a sequence of numbers, let's say on the top, and then you observe the output that is at the bottom. So what do you think is the function of this box F? Sorting, right? So the cool thing about is that this box is that it can do sorting, but how exactly it does sorting? And the fact is that if you anybody who took like data structure before, you have many ways of doing sorting. So the message I want to get through from this example is that even if we can replicate exactly the output, let's say we can replicate somebody's behavior completely, we are maybe unable to fully understand the logic behind. So that's one of the takeaway. And the other takeaway I want to mention is that like, what if like let's say if this F, you know, you fully study like in part one that you think is sorting. So how about like in this part that you have these numbers and what are the outputs? Do you expect that it is like also like sorted like before? We're not for sure because like the fact is that we build this model of F based on a particular set of values. And for here, the values at the range are completely different, and we are not quite sure what exactly is the output. All right, so maybe instead of like sorting, like it will do something else. So that's the other takeaway that we have to understand the data and the experimental condition that we are doing. That maybe in the lab, that when we do some experiment, it does not apply to, let's say, your daily life applications, or like if you have very particular tasks that you're doing, like it does not apply to like all other tasks that is not tested. So that's why it's really crucial to understand the set of like measures that you are doing, and also the conditions that you are studying. And then another example over here. So you have a series of sorted numbers, and when it comes out, it looks like something random, right? So what do you think is the function of G, right? So right now, I have to say we are not quite sure yet, because like the fact is that like this this box, you may need a longer period of time, so that you get an idea of what this box is functioning. And if you're just observing something in a very short period of time, you may not be having a very accurate description of what this system is doing. So I would like to quote a very famous statistician and George Boss, and he developed a lot of those Bayesian methods. And all models are assumptions. And assumptions, whether implied or clear stated, are never exactly true. And all models are wrong, but some models are useful. So the question you need to ask is not the model true, it never is, but is the model good enough for this particular application? So, so my message is that I hope to state is that like we always make our assumption, and we'll always make on like a subset, a sample set of data. So the mathematical model we always got this like limitation. But like for engineering, maybe for you too, like as long as we know its applications and it can like replicate like accurately for our purpose, then I think it's like good enough, you know, for our particular application. And the happy thing that I think like in the recent years is that I see a lot of collaborative efforts like between neuroscientists or like engineers, like because like we learn a lot from neuroscience on how to develop algorithms, and this algorithm in turn out help us to study the brain better. And at the same time, like when we have a better understanding of like the psychology of the brain, and we have better algorithms, engineers develop better like real life applications, and that have a positive loop. So I'm very like happy about the development in the recent years, and I'm very optimistic. So far, like in this talk, I have a general view of the field, like in terms of mathematical modeling for biological system. And for my lab, like we focus on three things. So first of all is the neuroscience, that is to quantify the neurological basis of cognition, and to better understand the neuromuscular coordination. So we collaborate with neuroscientists and also like those like in physical therapy or like in rehab science. So we study a lot of like athletes and musicians these days, and so try to understand like what makes athletes like run fast, what make a musician play music well, and we try to understand like what is the basis of cognition in various like situations, and also like we try to develop neuroprosthetics, not only motor prosthetics, but also we build models for cognitive prosthetics studies. And after coming back to Hong Kong, another research focus that I've developed, and we did way more these days, is that we try to enhance the human-computer interaction experience. For example, when I first returned to like Hong Kong, I joined like CityU, and then we built, I think it's already maybe 7 or 8 years ago, we built a multi-user like brainwave recording system at that time. So, but unfortunately, we haven't patented, but we try to use it during class to try to see like what's everybody's like EG during the class. So that was an interesting attempt. And the other one is that we build some of the assistive device that we try to do like study the hand gestures, and if we can use like simple wearables, like just vibrations on the wrist can already tell the hand gestures that we are doing. And we also work with like industry, for example, Intel, so to develop better AI algorithms based on what we learned from the neuroscience. So I think I will stop right here. So hope that I'm doing good in time. Thank you.
Thank you, Rosa, for giving a very comprehensive overview of the state of the art of brainwave technology, hardware, and modeling methods. So again, we will open the chat room in Zoom for a question from online participants. So please type in your question for Rosa. At the meantime, again, I will start with my question. So you have mentioned, because when I was reflecting on how this technology can be applied on artistic purpose. And I recall in the past when I experienced artworks that were using, for example, EEG technology, many of the artwork that I think are a bit of improvising the measurement instead of, the artist may be interpreting the measurement with a artistic concept or expression, rather than yeah. But as you also talk about the recent achievements that scientists have made, do you think the state-of-the-art hardware and computational tools are accessible and powerful enough for artists or other practitioners to use those tools to do some meaningful measurement?
So far for EEG, there are a series of research, and the most used results was on sleep, or like the arousal state, and also like more on clinical like application, for example, like seizure, like so. But like in terms of the other applications, time by time, like it is a sort of like experiment constraint. Like for example, like you may recruit like 40 people. Like you may see a lot of work that is like recruiting like a subset of some people, and then I try to like see for this task, like can I map from certain EEG features that help to decode like from EEG to that task. So that's what I observe. So I think right now the tools is becoming much better in terms of also using like it for like identifying like different like motion preference or like some of the emotion studies. But because like every single body like is being like varied quite a lot. So what I observe time by time in terms of EEG application, we have to be careful about the like artifact removal, like or else like basically your eye blink, your talking, or like even your head move already like override all the EEG signals you measured. And also like even like you did that, you have to be aware of the inter-subject variation. So time by time, actually, if you really want to use it for like a real life application, you need a little bit recalibration. So instead of like, it's very difficult to say I can use a general model and apply to everything. So, but I think like right now we start to have some open source tools that is like sharing in the field too. So, but I think for EEG, like it's about like this is sort of one measurement, but I'm more concerned that it's about the experimental design and also some of the pre-processing. Yeah.
So I think we can open the Q&A session to the floor, to the participant physically here.
My question is, Moore's law is really dying.
Sorry, I don't hear you clearly.
You mentioned Moore's law. So don't
Yeah,
you think Moore's law is actually dying because the number of transistors not doubling?
Oh yeah, so that is a interesting question. So previously, like I still remember Intel in 2011, they showed a slide in their stock whole like investor like what will happen for the trend like after like 10 years, and they literally say they have no plan. So. But right now, we can see that it's still like catching, but we are reaching some of the fundamental limit in physics. But I personally, I'm sort of like optimistic. So right now, people can use multi-core or like different strategies to try to overcome this like limitation of physics for one single chip. But overall, like the key point is that the cost of making or doing similar calculation is much lower, and it's like much faster. Like overall, the trend did not change much. Yeah.
I have a follow-up question.
Yeah.
What do you think should be the lowest size a transistor could go before quantum tunneling stops it from shrinking even more?
We're actually reaching the limit already. So but at the same time, like people like start exploring like quantum computing. I know nothing about quantum computing, unfortunately, and right now like most of our architecture like right now is still using the sort of the von Neumann architecture. So when you're talking about like quantum computing, I'm not quite sure if like all the tools that we have can be directly transferred to the other kind of architecture quite like immediately. But so far, like I just have to keep the belief that the cost is lower, and overall, like we can do the same computation much faster too. Like given all these trends throughout the years. Yeah, but like if you talk about like fundamental physics, we cannot change. Yeah.
Okay. My second question would be, you were talking about some neural prosthetic limbs.
Right.
So when do you think their cost will go down enough to go to the general public?
Oh, that's actually for the prosthetic limbs. You may see some of the DIY attempts, 3D prints, and like the code from the stumps, and to the prosthetic limbs from SEMG. So that is the electrical muscle activities, and then for that part is actually the cost is can be quite low already. So. But like for like some of the cases that is relate to the limbs, the lower limbs, or that you need like stronger, like better motors or like better like design, like hands, etc. Maybe that may cost higher. But like right now, I feel the trend is that it is going much lower already. Yeah. Yeah.
You've had 3 questions. I'm going to ask a question now.
I mean, one was a follow-up.
Yeah.
Okay, this is the last one.
Yeah.
Could you just go over once more about your sorting thing that you were talking about? That's the last thing. The sorting thing, the sorting function.
Oh, what the start?
In the presentation?
Yeah, in the presentation.
Yeah, the function f.
The function f. You
Yeah.
mean this one?
Yeah, so were you talking about normal sorting functions, or were you specifically talking about something that uses machine learning in that sorting thing?
Sorting. Oh, yes. So like for like data structure. So for like most of engineering students, they have to take a course called data structure. So in short, like it's like how we have to learn about algorithms. How you organize your data, how you search things. So one of the items that we have to go through for all the undergrad engineering students is like how
first
yes. So there's a different way of searching. Just like when you are organizing your wardrobe or your bookcase, everybody has their method of sorting. Some is like divide and conquer, some do it in sequence. So what I mean, what I want to use this example is like try to like bring an analogy to brain modeling. That is, even if we replicate the same output, for example, even we can sort the same thing achieve the same output, but like what exactly the sorting method we used is like difficult to tell unless we probe even deeper inside to this box F. For the brain, like we may need more measurements so that like we know exactly what the brain is doing when it like give a certain behavior. Yeah. Thank you.
I have a more philosophical question.
Yeah.
It may be incoherent, but I'll try and ask it. It's it's very interesting hearing you talk about your work and how you're proceeding in a very, as an engineer, in a very practical way. A lot of cognitive science models that philosophers and cognitive scientists develop of the brain. They're very ambitious, holistic models of how the brain functions. And of course, empirically speaking, our knowledge of the brain is still very rudimentary, and it's probably going to be hundreds of years before we achieve anything like an empirical knowledge of the brain. So given that, I wondered whether for you, for an engineer, it's better to think about our theorization or modeling of the brain in terms of, in a piecemeal way, rather than seeking to sort of look for a holistic theory or holistic understanding, that we develop understandings of the brain according to the specific problem that we're seeking to address. Like, for example, sensory perception, and we have one model of how the brain works in order to understand that, or understanding limb movements, and we have a different model of the brain, and we shouldn't even expect that those models necessarily cohere into one single model, because our understanding is so rudimentary and the brain is so complicated. I mean, we might in the future, 50 years or 100 years down the road, get that holistic model. But do we actually need it as research scientists, from your perspective?
So, excellent question. Sometimes I think when we try to simulate the behavior, like let's say it's good to one of the audience member mentioned a little bit about architecture that raised the discussion. So sometimes I think like for our brain, we exist in a biological form. So if they have to simulate something, it's actually analog simulation. Maybe like we know each neuron, how it works quite well, actually maybe building a biological simulation may be more efficient than using a digital means to simulate how the brain works. Right now we are, that's why I mentioned about that that quote at the end, is because because of the limitation of the tools we have and the assumptions we have. So that's why, like right now, we're sort of doing it part by part, like for our application. But like if we are going to like do a more like complete like simulation, maybe in that case, I think in some sense analog simulation or like biological simulation, and that kind of architecture will be more possibly more like suitable, like than the digital means maybe.
Thank you for a very interesting talk. As I'm listening to your talk, I tried to imagine if I was called upon to present artistic process to a group of engineers and the difficulties I would encounter to do that. My question is related to the one just previous, hopefully not totally overlapping. I'm also interested in the aspect of your research, which is the quantification of cognition. And you talked of a model where there's an input and an output and a black box in the middle. I'm wondering where, for the purposes of your research, you would define the output of cognition that you're looking to quantify. Is it behavioral? Is it, how is that defined?
Oh, so for me, it's like completely application driven. Let's say my clinical partner, okay, they want me to like to understand the motion, from like let's say from brain signals or muscle signals, peripheral signals, to like hand gesture, like motions, like so the output is then let's say the kinematics of the hand, of the limb. Or like our output can be also, let's say, in terms of rehabilitation, let's say how like this particular patients will be likely to have a faster or like shorter rehabilitation time to restore the same function. Or we could also have like emotion, like for example, traditionally we have some like so-called the VA model, the valence arousal model. So this can be output to, and then we study based on different images, different audio stimulus. Like we recruit a lot of subjects, and like they have to like evaluate like all the images and audio they hear or they see in terms of these VA levels.
So this is what would determine that cognition has happened?
So for me, like right now, I'm more focused on the application side. For the cognition, like we also had previously some memory-related like tasks too. So yeah, so I've misunderstood that you would like to know what our lab has been really doing. But like for memory, we did like some studies, but not on humans, but for animals. That they have to do certain tasks that show that they memorize something. And then we show that like using certain deep brain stimulation that generate from the models can restore that kind of like short-term to long-term memory encoding function. Yeah.
Any other questions? I have one more question for you, Rosa. I know that your group has also been using deep learning approach to understand the measurement that you have obtained from the brain. My question is, because most of the time when we are using deep learning approach, we need to have ground truth data.
Yeah.
How do you get the ground truth data while you are, yeah.
Oh, okay. So for example, let's say, so there's some, like our group also opened, like shared a lot of like data sets. So for example, we measure, let's say from the data set that is like from like brainwaves, EEG, to like, let's say like motion, or like brainwaves to like emotion, so VA model, etc. So all these, there are some like data set that is collected from experiments. For example, using one measure is like, let's say the emotion experiments. We present, let's say, the images or audio in parallel. Like we measure the, let's say, the EEG, and also the evaluation of the subjects in terms of what they feel, perceive as the valence level and the arousal level, the emotion they perceive from certain image or audio signals. And then from there, we'll build that kind of like input-output data set. And then like we can use like deep learning techniques, or like, of course, like we usually the limitation for biomedical applications is that like we only have a very small data set. You know, it's not like when like computer science talk about, oh my God, there's like so many thousands and tens of thousands, or maybe even like for Google, maybe millions of like images, right, for them to analyze. So we are working with a very small data set, very noisy data set, and so that's why time to time when you read the papers and the performance level varies. And when we talk about some certain tasks that is like really subject dependent, for example, emotion, another problem that you may encounter is that everybody like describe the emotion quite differently. Yeah, and then so that sort of like make your data, you know, varies a lot, like. And so that's why it could be challenging, like when you are doing modeling. So sometimes like we don't use like deep learning for some of the data set that in some sense like we use some simple method is sort of sufficient. Yeah.
Okay, thank you.
Yeah.
Am I allowed to ask a
Of
question?
course.
It's a very, very stupid question. So Rosa, you mentioned in the passing, one of your research projects is to study musicians, right? So in fact, I'm surprised nobody kind of asked this. So I'm really, really curious what kind of to know more about this project, to be honest. Like, what's the goal of this project? Is it to kind of make people play music better? And if you can study musicianship or musicians, can you use similar your experience to study actually more abstract things like love and emotions and?
For this musician project, so I collaborate with Dr. Vincent Juran of biomedical science in CHK, Chinese University of Hong Kong, and he is a pianist. So we have been collaborating in other rehab projects, and then I turned out him and some of his colleagues are also very good pianists. So we are like, oh, we have been studying how the muscle work together so that we can help people to rehab, right? So can we study the strategies that musicians used so that we identify, okay, something quite abstract, I have to say. So usually when I play guitar a little bit, but I sit in a master class, and then I remember it's a Pepe Romeo. Is he said like feel the string, and I still have no idea what was it. Feel the string, you know. So when people teach about I feel like for music, I don't know if it's for art, but they usually use very abstract terms. What we try to do is can we use a more technical or quantifiable terms? Let's say for somebody when they just maybe need more muscle training, you know, for certain parts of their body or certain finger training to enhance their finger independence, so that so that we know different like musician styles, etc. So it's still ongoing. So if you are like for the audience over here, if you are a pianist, like contact me. So we still need a lot of subjects.
Wouldn't that take the magic out? Like I think feel the string is good enough, right?
So that's why like this is like feeling is a little bit disturbing to engineers, you know. We like solutions. We like solutions. It is very frustrating for us. Yeah, I know. But that's why, like, you see, the feelings is a problem for engineers.
All right, thank you, Rosa. So it's time for the third speaker of today. Let me introduce him. He is Dr. Yun Hua Lam, who was the last person who asked question for Rosa. Yun is a biochemist and cell biologist. He was a postdoctoral researcher in Wellcome Trust Biocenter in Dundee, Scotland. He is now an associate professor at Department of Chemistry, City University of Hong Kong, where he built a multidisciplinary research network to tackle a number of biological problems, from environmental sciences to regenerative medicine. He has published over 100 scientific papers and patents, and is currently the leader of the Global Research Enrichment and Technopreneurship program at CityU. He was a scientist in residence at Symbiotic A in Australia in 2019 and the recipient of the CityU Innovative E-Learning Award in 2020. He is a co-organizer of the Leonardo Art and Science Evening Rendezvous Laser and Café Scientifique in Hong Kong. He collaborated with Maro Pebble on the artwork Microbial Emancipation in 2020 and is the scientific advisor to a number of artworks, including Magic Wands, Buttons, and DNA Splicer by Wong Kit Yi and CRISP Seed Resurrection by Ken Renato in 2021. The title of his talk is Life is a Mess, Towards a Gene-eccentric and Post-teleological Discourse in Bioart. Over to you, Yun.
Thank you very much. I kind of sent this biography to you as a joke, right? I didn't know you really read it out. So thank you very much for inviting me, and it's an honor to share the stage with David and Rosa. So all the important talks of this section has been given. So if you are not interested, it's dinner time. So basically, you can relax a little bit. So you can share my slides. So basically, this talk is just a discussion. Life is a mess is a summary of my life at this moment. So. But I'm a scientist, so I'm different from the previous two speakers. I'm not an engineer or a machine learner or machine learning researcher. So the kind of the style of the direction of the talk, or the kind of the nature of the discussion will be changed, right? Or the kind of level of complexity will change from a kind of a computer mathematical model problem to a biological question. In particular, I would like to kind of discuss a new trend of how art researchers and philosophers are now looking at biological questions, right? So basically, here's the quote I would like to share with you, kind of to set the stage of the talk. So it's basically Richard Dawkins. He wrote a book. If you kind of didn't know, he's the author of an extremely influential book called The Selfish Gene, right? And so basically, he in one of his books, later book, he wrote, what lies in the heart of every living thing is not fire, not warm breath, not something called spark of life. Instead, it is information, words, instruction. If you want to understand life, don't go about studying the vibrant, throbbing gels and oozes. Think about information technology, right? So basically, 2013 is the year when the Human Genome Project is complete, right? So the first draft of the Human Genome was completed in the year 2000. So, but then it was the first draft, right? So there are lots of gaps that hasn't been sequenced, but then these gaps are closed in the next decades or so. So by the time when he wrote this sentence, basically the entire human genome has been sequenced, right? So the background of the slide is basically a snapshot of our own genome, right? Kind of with all the genes and every single letter of the genetic code has been sequenced, right? So at this kind of in an era like this, it's very, very tempting for a philosopher to think that we have hacked biology, right? So we know all the data, and therefore we can forget all the messy stuff in life and in biology, and focus on the information bit of biology. So I would like to devote this talk to the discussion of these two sentences. So my plan is, actually, I don't know what to think of that. So kind of this talk is a little bit like kind of a preparation for a discussion that we're having. I would like to divide my time into three parts. The first part is I would like to introduce about the historical background, about why Richard Dawkins and many other thinkers are starting to think of biology as an information question, information subject, discipline these days. The second part is to tell you that why I think, as a research biologist, that this is actually not true. It is not possible to think about biology and life as an information technology. And I think that echoes the discussion we were having earlier with Rosa about kind of how much you can model something, right? So, and then the third part is that I'm going to talk to you about some of the work we are doing in our lab that's related to this discussion. However, I tend to speak too much, right? So basically, I overrun all the time. So I'm happy to stop at any point in order to have a more discussion. So biology as an information technology, right? So I think everything started from this idea, right? The so-called central dogma, which is basically an idea of how genetic information flow from DNA to proteins, right? So basically, for the non-technical, non-scientist people in the audience, don't worry too much about these jargons. But the idea is everything in life is actually constructed, and all the functions are carried out by a family of kind of biochemicals called proteins, right? So, and kind of proteins make up our cells and kind of enable all the reactions that is happening in life. And the information of all the proteins are contained in something called RNA, and the information in RNA is contained in something called the DNA, which are the genes, right? So basically, central dogma states that this information flow only in this direction, from DNA to RNA, from proteins, and somehow once you get to the proteins, and you will get life, right? So basically, this is what kind of biologists believed since maybe the 1960s. So, and the interesting thing about this is how the information flow is actually controlled by proteins, right? So basically, it's sort of closed the loop, right? So how active is a gene, or so-called whether a gene is turned on, is actually controlled by proteins from other genes. So you suddenly have this kind of a self-contained or self-organized network of functionality that is kind of really, really suggesting or point towards that the genetic network inside our bodies is like information network. So, and so basically, and also, and one more point is, scientists believe, although some new data shows that may not be always true, that the information of the proteins and RNAs are always contained in the DNA, right? So it is no longer necessary to write the word RNA and proteins, because there's a kind of redundancy of information, right? So once you know the sequence of the DNA, you already know the sequence of the RNA and sequence of the proteins, and therefore everything about this protein, right? So therefore, scientists, we are either very smart or very lazy, so we use shorthand, right? So the shorthand is this kind of the universal biological shorthand for this diagram, right? For this diagram. So basically, this line is the DNA, right? The DNA is active and making something, right? And then this kind of activity of the DNA is being controlled by something else. You probably can guess that the arrow is the protein, right? So the line is the DNA. When you have a short form like this, it's starting to look like an engineering diagram, right? Basically, the engineer look at this and, okay, I get this. So in fact, the genetic network, or the so-called gene expression regulation, in this case, actually human genes, really can be narrated or described by this kind of engineering network diagram, right? So once again, a line is the DNA, an arrow is a protein, and the vertical arrow is the regulation of this activity of the DNA, right? And you can see that the one gene is being regulated by another genes, and then here the brown words is actually a hormone, right? And then you can also notice that these arrows can be imagined as an output, right? Everything look like a computer, right? So basically, and then in fact, you can actually rewrite a biological textbook by using this shorthand, right? So it looks crazy complex, right? But you can now, even for non-specialists, start to understand, right? A line is the DNA, an arrow is a protein, right? Vertical line is the regulation of this DNA. So now, if you kind of put this together, and by the way, if you still remember, activities of one gene is regulated by the product of another gene, right? So this is exactly what's happening. This gene is regulated by this gene, right? This gene produces the green thing becomes a vertical arrow regulating these genes, right? And then this gene regulates this gene regulates this gene. Sometimes the regulation of one gene is a combined effort of two genes. So now you can start to appreciate how much this diagram, this example is a genetic regulation of a bacterial network. You have input, so basically these are nutritions, right? Nutrients, concentration of nutrients. This is output, right? This activity of one gene. In fact, you can actually see that this kind of semicircular thing, I'm sure Rosa can kind of tell us that it really, really look like a circuit logic gate diagram, right? So in fact, this is actually regulation, the activity of one gene is regulated by combined effort of two genes is an AND gate, right? So now if you kind of starting to think about that, then a bacteria or genetic network is a computer, right? So the bacteria is sensing all the inputs, for example, concentration of nutrients, and then giving out output, right? The output may be to die or to divide, or maybe to express some particular genes that we can measure. So, in fact, MIT, the organizer of an international synthetic biology competition called iGEM, they stopped calling genes genes. So instead of calling them genes, in fact, not just a gene, but also the regulatory element of a gene in a kind of a self-contained cassette, they stopped calling that genes, but they kind of use another new words, right? And the word is called biological parts. Genes are no longer genes, but they are parts of a machine, right? So in fact, you can go to this website. It's called Registry of Standard Biological Part. Anyone with an email address and a credit card can buy biological parts so that you can assemble this part by putting them into a bacteria and make the bacteria do the things you want the machines to do, right? Just like making a transistor radio and something like that, right? So anybody with an email address and a credit card, for example, can turn your bacteria into any color you want, can do anything you want, GFP, different fluorescent proteins, you name it. So the next kind of leap in this machine kind of analog for biology is that since we know all this part, right? So since we know all this part, so instead of modifying existing cells with an exogenous part, scientists can even make an entire genome, create entire genome simply by chemical synthesis, right? So this is a paper by Craig Venter, the Elon Musk of biology, right? So basically, whatever Elon Musk is doing on Mars or neuroscience is doing in synthetic biology. So what his company, interestingly, is not a university, is not a research lab, but the company is challenging is that they can create life from molecules, right? So, so what they did is they will create a genome, basically chromosomes, right? Because they know the function of all the genes, right? Anyway, so they can actually create a genome by using 4 bottles of chemical, right? There's 4 letters of DNA, and they can actually do almost like an organic synthesis, right? Basically, is a synthesized chemical, which is DNA, totally based on almost like a digital description of life. They can kind of turn this digital information into an object, and they can put this object into a cell, and then they can make this cell work, right? There are actually a lot of very, very interesting things in this on this slide, but hopefully we can have, please ask me a question about this slide during the discussion time. But this is the outcome, right? This video was actually from a paper published 2 months ago. I look at this video and I still, to this day, kind of feel this is a really, really amazing and important video. So what they're doing is they do this digital DNA synthesis, make the DNA in a lab, and they can actually create a cell. Here are the cells, and the cell will give fluorescence, and the cells can eat and divide in just like a bacteria. So this is really an achievement. And now, because you can create life from 4 bottles of chemicals, and it works, right? So you can see from this diagram, it works. So now the next step is, because I know everything about a cell, right? So basically, I know everything about a cell. I can digitalize the cells, right? And in fact, I probably don't need the cell, right? I can even upload all the information on the computer and build a so-called whole cell model, right? And I know everything about these cells, and then I can actually input whatever information I want, concentration of nutritions, molecular interactions, and then I can measure the output, right? And because this model is based on everything we know about the cells, and we seem to know everything, so we don't need a cell anymore, right? So this is kind of mind-body discussion all over again, right? After hundreds of years, we are still fixated on this idea or possibility. So until basically now, right? So Hirari, kind of superstar historian, he even said that all organisms, in actual facts, are algorithms. Human beings, giraffes, viruses are all algorithms. They are different from computers only in the sense that they are biochemical algorithms evolved at the whim of natural selections. So basically, he said that in 2018, but then this idea of humans or body as a machine has been our fixation for decades, I would say probably for centuries. So we are kind of still coming back to the same starting point, right? So it comes to the fact, it comes to this, and the idea is our body is a machine, right? And the machine is well designed, right? Designed or evolved through billions of years of evolution. So every part of our body has a function. So the classical example of this is our ear, right? So basically, our ear has a really, really interesting shapes, right? And the idea is these shapes will direct sound into our ear, right? Basically, so that we can collect sounds from all different directions, and we can also sense the direction of the sound, right? So basically, every bit, our ear, the design of the ear, just like a car or a mobile phone or any other machines in our household, every part has a reason. Every part have a function. So, and this is kind of the state of the art now, right? So, however, a lot of people ask, we basically know the function of every bit of our ear, right? So, except for one part, and this part is this part, right? This part, what's the function of that then? So it has no bone, right? It's doing nothing to direct sounds into our ear, right? So why does it exist? Why didn't evolution eliminate the ear knob? Why didn't evolution eliminate something useless, right? So in fact, as a historical footnote, the function of ear knob has consumed many careers. So because everything has a function, and we couldn't find the function of an ear knob, so scientists are unhappy, right? It's like feelings is not good for engineer, but lack of function is really, really not cool for biologists. So this is from Wikipedia, right? Ear knobs have no bones, right? But a lot of blood supply. That's a fact. If this is true, if it is true, then there must be a function, right? So the function must be to keep warmth, all right? But then there's no evidence for that, right? So finally, the function is ear knob must be an erogenic zone. So the function of ear knob is to make us having better sex. So there has to be a function, right? And we're coming to the kind of second part of the talk, right? What makes biologists so obsessed with having this functional body idea? So we believe it's because the way we learn biology at school, right? So this is actually from a textbook, not textbook, but internet something teaching learning thing. So it's about evolution, right? This is how we teach our kids evolution. So basically, there are two kinds of mice. One is black and one is white or light color. Eagles can see the light color mice, but not the black ones. So the light color mice will be eaten. So the black ones are there having all the sex and reproduce, right? So that's the kind of a simple, almost like schoolboy description of evolution. Everything on this slide is wrong, by the way. But then anyway, that's how we learn evolution, right? But then if evolution is so powerful, and kind of after billions of years, all the features in our bodies have been selected by billions of years of evolution, then maybe we can actually condense this sequence by this slide by saying that the function of our skin is to avoid ego, right? But then it's of course not true, because this the skin color of a mice may be controlled by hundreds and hundreds of genes, right? Each genes are only playing a small role, and the role is really, really undefined. Just a historical footnote, scientists actually find out the genes for knobs. So it must be selected by evolution, and therefore there must be genes, right? And scientists actually find the genes, right? So this is actually very interesting paper. Once again, I don't have time to go through the science and the meaning of the diagram. Please ask me during the discussion, right? But this, they found that the size of knob is associated with 4 genes, right? So basically, this one of the genes, this one, is on chromosome 2, right? And I couldn't go into the technical detail, but then the evidence showed that this chromosome 2, there's a kind of a gene in the chromosome 2, is really, really associated with the size of a person's knob. So, but then that's kind of a correlation, right? It's not a cause and effect. And nobody knows why these genes can change, can make somebody's knob larger. In fact, this paper, this gene, this association, this genetic association is only true for Latin Americans, right? And the same gene for Asians, for us, has no correlation with knobs. In fact, the same gene, the same kind of position in the gene, so-called gene locus, in Asians is associated with how hairy we are, not how big our knobs are. So basically, this kind of gene function association, we are currently knowing so little of it to say the fact that we can understand the whole thing. So the second thing I want to point out is, so we are looking at, we can sequence a lot of genes, right? And we basically can describe evolution in action, right? So this is the data of COVID-19 virus from February last year to now, basically to this data is good. I think it's up to date to last Friday. So basically, they sequence. So every time there's a spot, there's a sequencing, right? They sequence the entire genome of the virus, and you can see that, wow, this virus is actually starting from this point, and then it diverges, right, by doing this mutation, right? And we are talking about these genetic variants of COVID-19 a lot, and some variants are more dangerous than others, but then, of course, some variants are more infective, makes the virus more infective than others. But most of the mutations, for example, this one, they found the sample in Slovakia, and these are the mutations, right? Most of the mutations, I would say 99.99% of the genetic variants of the virus has no effect or no observable effect to the infectiousness of the virus. We didn't know what happened to. We didn't know the function of this mutation. In fact, each mutation happened inside the body of one person, tells a story about how this virus was adapting to the body of that particular person, responding to the environment, immediate environment of that person. This is an epical story about this virus at that point of time inside that person. But without all the information, there's no way for us to know the so-called function of this mutation. I guess the point I want to say is each mutation or each DNA sequence that we are finding tells a story about a specific evolution or adaptation event, and without knowing, and then in retrospective, it is impossible to retrace this story. Therefore, all these mutations or genetic variations are like some kind of personal memory of this particular species or this particular cell that is only meaningful to that cell, but not to the general so-called function of the organism. So I guess I don't have time for this. The third thing about biology is we know the genes and we know everything. But do we really know everything? So this is a very interesting thing, interesting topic, right? So, and the topic is called emergency, something that I'm sure a lot of you are interested in or maybe even working on. When a system is becoming more and more complex, it becomes so complex that it is impossible to be modeled. It becomes so complex that it's impossible to be modeled unless, at least at this particular moment, right? So, and biology is one of these systems. So we know everything about one gene, right? So once again, this arrow, unfortunately, this arrow in this diagram points to a different direction, but an arrow means the protein, right? So horizontal line is the DNA. So the activity of this gene, right, basically can be measured by the output of the DNA to RNA to protein, right? This is exactly, this is still the central dogma. But this output is actually not just controlled by this simple gene, RNA, protein journey, because this concentration of this protein is actually influenced by many, many other proteins, basically, and so on. And each of these proteins is independently influenced by its immediate environment. So basically, the whole thing is so complex that we now know that it is actually impossible to predict the outcome of a gene expression. So about 20 years ago, and it was only about 20 years ago when biologists have the technology or have the methodology to visualize the output of a gene, meaning the concentration of a protein that is being made in individual cells accurately and in real time. In individual cells accurately and in real time, right? So for example, this is actually a dish of bacteria. There are actually a lot of bacteria in this diagram, but trust me, right? There is not visible now. Whenever this bacteria make a gene, the bacteria will become white, right? So when I play this movie, this bacteria has received an external stimulation, right? We know that this bacteria will respond by making this white, a gene that will emit white light. So all models suggest that you add this stimulation, then all the bacteria, because we know everything about this gene, will make this white protein at the same time, right? So all the cells will become white kind of progressively. That's the prediction. But when biologists can visualize this expression of the genes in real time, they saw this. So this cell is becoming white, this is becoming white. But then they turn off the genes, right? Some of the cells become white, but then other cells will become white for a moment, and then they turn off. Some cells become whiter and whiter and whiter, and kind of. But then other cells will actually turn. Instead of having a predictable increase of concentration of this protein output, these cells will turn on and turn off the proteins all the time. it is kind of it is kind of like you have a lot of computers, and then you switch on all the computers at the same time, but then the computers, they don't switch on all the time at the same time, right? In fact, some computer will switch on, and then some computer will be switched on 3 hours later. Another computer will be switched on and then turn off again, and then turn back on again. So what's going on? It turns out that this idea, in fact, Rosa mentioned this word before, right? It's called stochasticity. It turns out now we know that you can predict how the possibility of a gene being turned on in a cell. You can predict the possibility, or you can predict the percentage of cells expressing that gene in a large population. But you can't accurately predict both things at the same time. So, so basically, this is interesting, right? So just one more point before we finish, and I have got to show you this because it's really my favorite movie. So basically, the previous movie was in a bacteria, right? Bacteria responding to a changing environment, but this is now human cells, right? So maybe human cells may also be doing the same thing, right? In fact, this movie is more or less the same as the previous one, but number one is in human cell. The second one is that gene is not responsive to an external environment. It's not kind of responsive to the concentration of a kind of a nutrient we put in the culture medium, but it is actually a so-called biological clock, right? So basically, these genes that we are looking at will be expressed at night, right? And then during the daytime, it will be turned off, right? And basically, it's an internal biological clock, and we know a lot about these genes, right? And what you're seeing is a dish of cells, and trust me, there are possibly hundreds and hundreds. I think there are probably 500 cells in this diagram. And whenever the cell express these genes, it will be you will see white light, right? Light. And what you're going to see is the scientists in this paper start to image this dish of cells in daytime, right? So this level of gene expression is very weak, and then they will image this dish of cells throughout the day, right? So the prediction is daytime, this gene is not expressed, and then during nighttime, right, then all the cells will start to make these genes at the same time. So, but instead, the idea is now daytime, and then sunset, right? Evening, basically nighttime. Instead of having all the genes, all the cells turning on this gene in a programmable, quantitative ways, what we see is flashes of light. You can see cells almost like randomly turn on and off these biological clock cells. So in an unpredictable manner. Towards the end of his life, Alan Turing, the father of modern computers, in fact, the last paper of his life is actually about biology. He, being a genius, he really wants to understand how a biological system can evolve such precise and beautiful patterns. So the end result is Turing pattern, right? So the idea is, without a shadow of evidence, in his ingenious mind, he came up with a model of two diffusible molecules, right? And the cells respond to these two molecules by making black cells and white cells, and then these two molecules will mutually influence each other, right? And then one of the molecules will inhibit itself. By changing the diffusion gradient, diffusion coefficient of these two regulators, and the inhibitory coefficient of these inhibitors, Alan Turing said he can create any model, any pattern in biology, or in fact, any patterns. So it's great, but for about 50 years, biologists has been looking for real-life example of biological systems that followed his idea. And I think they found about one example, and basically it's not universally true in biology. So the Turing pattern is beautiful, but not great, right? So until scientists in 2018 factored in stochasticity, then everything works, right? It turns out this Turing pattern, for this to work, at least in biologically relevant concentrations, the cells has to respond in a imprecise and unpredictable ways. So really the last bit. So basically, really looking forward for the discussion. But what I wanted to tell you today is, at least based on our data now, information now, is far too early. I think we are centuries too early to say that life can be uploaded to a computer, life is a program, life is information science. In fact, I think that biology is not gene-centric, is not programmable, is not so organized, definitely not functional, and not precise things enough for us to model, at least for the next few centuries, for the next few, not century, for the next few decades, maybe ever. So once again, thank you so much for your time and attention, and looking forward to the discussion.
Thank you, Yun. Whenever I listen to your presentation, I feel your energy and passion to the science and research that you are working on. So it's time for Q&A again. We will open online in the chat box for online participants to type in their question for Yun. You asked us to ask question and about that slide. Do you want to explain that slide to us?
Which one? So basically, it's the Craig-Venter synthetic genome, right?
Right.
So that's really a this is a really interesting work, right? So I kind of told you about this first bit, right? So scientists, they always want to paint a picture that after knowing all the genes, at least in bacteria, so in fact this is actually a bacteria. So basically they know all the genes in bacteria, and they can even judge which gene is essential and which gene is there, but then they're not doing so much, right? So I'm kind of the human equivalent to that non-essential genes. But then E. coli have 4,000 genes, about 4,000 genes. But what this company does is they decided only 400 genes out of these 4,000 are really essential to make this bacteria alive, right? So they just eliminate all the other genes, take these 400 genes and paste them together in a very small piece of DNA, put it in a cell. They call it rebooting these minimum genes, and it works, right? It works. It works, meaning that it is a self-sustained, free-living organisms and divide, right? But what they didn't say, they said, but then what the generalists didn't say is that to make this synthetic genome to work, they need this, right? This blue thing is actually a cell. What they did is these cells have a chromosome, right? The white thing is a natural chromosome of that gene. This blue thing is the synthetic DNA, right? So what they did is they will put this thing in this cell. Now this cell have 2 chromosomes, right? And the interesting thing about the experiment is these blue genes, the synthetic DNA, contains an enzyme that come out and digest the original host DNA, leaving behind nothing, no genetic material except for the synthetic DNA, and the synthetic DNA will go on and do its magic. So to me, as a biologist, it's got this neat information, digital information, but then the kind of the so-called messy stuff. They need the messy stuff, these blue cells, to, in fact, to interpret this genome and turn it into something functional. And while this DNA is completely human synthesized and completely understood, but what's happening in this blue cell, we know very, very little about it. So in order to make a synthetic life, we still need a little bit of magic, right? Still the messy stuff, these oozes and ribrins and throbbing gels. And this impossible to be ignored. So that's kind of that's what we I wanted to say for this slide.
All right, thank you. Question from audience.
Yeah. Thank you so much for your interesting and great presentation. I have a question, maybe a little bit in relation to the previous talk, and of course in relation to your talk. So I'm interested in the function of model organisms in biology in relation to the messiness of life, as you explained, because I really also liked that quote from George Box, which was mentioned in the previous talk, that all models are wrong, but some models are useful. So what is your opinion? What is your opinion about biological model organisms?
Would you like Rosa to answer first, and then me, followed by me, or?
Maybe you first.
So that's is a very, very interesting question. I think some philosopher or art researcher should write a book about biological model organisms. As a professional biologist, I was taught to don't waste my time on useless organisms. Only work on a few so-called model organisms, right? So, for example, for fish, we have zebrafish. For bacteria, we have E. coli. For fungus, we have baking yeast. For mammals, we have mouse, so on and so forth. Insect, we have Drosophila. In fact, almost everything we know in biology, we have millions and millions of species, but almost everything we know about biology is based on fewer than 20 organisms, right? And the reason we did that is because we have limited time and limited grant money, and therefore we want to focus our resources to understand a few organisms really, really well. There's a famous quotation by, I think, the most brilliant molecular biologist in history. He said, "What is true for bacteria must be true for elephant." That's the idea, right? In fact, he said this during his Nobel lecture. So he got a Nobel Prize, so he must be good, right? So, so the direct consequence of this idea, instructed by this great men because they're all men anyway, is that we should focus on so-called model organisms. However, now the kind of indirect effect, but you can probably guess, is we know very, very little about the other millions and millions of species. So therefore, we are now in a dilemma, right? So Rosa sent me something yesterday saying that some butterflies have transparent wings.
Yeah.
Right. So that's really, really exciting, right? Butterfly, they're not supposed to have transparent wings. We have a lot of exceptions and kind of weird and queer things in biology, and we don't know anything about. So, you want to say something?
Yeah. First of all, I hope that I understand your question correctly. So, but for me, like for modeling for biological system, so of course there are lots of diversity, and then so I would say fundamentally, so it's still like DNA, RNA, protein. Some of the fundamentals are the same, sort of like the brain fundamental is still neurons, but every part of the brain they have different structure. And then a little bit of different things, then it contributes to different functions ultimately. So I think when it follows certain distribution, I have to say, follows distribution, it will have certain patterns that evolve. So, I think biology is a really, originally I'm 100% engineers, in my training. And then gradually, when I learn more about the brain, I learn about biology, I feel more I know nothing, I feel like. But I think some of the terms, in terms of the mathematics or biology, actually can be explainable in function in some sense, even though you feel like stochastic is not, why is it a good thing, right? But for stochastic, if you take a look at in terms of a time scale, if it's a time scale, actually it reduces energy. Some of the stochasticity, actually it reduces energy. And if you have a large scale system, actually this probably can help in terms of the robustness of the system. So I think some of the biological system, how they behave, maybe in some sense, when you feel like it's it's stochastic, but sometimes it may actually stochastic leads to a certain purpose.
But then it kind of follow the same track, right? You have to have a function.
Well, I don't know. Can I ask a question? Yeah, two. All right, okay. So I have this question then. I don't know if it's appropriate, but there's engineers, that question has been in mind. So for certain culture, for example, we talk about evolution, right? We are not talking about yellow because it has been there, but for certain culture, they on purposely do some surgery to remove certain parts in their culture, But then after all these years, they still have that body part, right?
Yes. So you're talking about foreskin, right?
I don't know the biological term, but yeah. But then,
so the question is why are foreskin still there after being after circumcision has been around since pre-biblical times?
Yeah.
So basically, that's so-called. So I'm just I have so many jokes coming out, bubbling in my head. I'm kind of suddenly stop responding. So basically, the removal of foreskin doesn't affect our kind of lifespan or reproduction. I think there's some. So why? So basically, just like so basically, removal of a body part after 1,000 generation won't destroy this body part, right? It's not how evolution works.
Yeah, because usually when we update system, all right, so it's driven by error, or negative, or that triggers certain pathway that update something. Yeah. So
we haven't got much time, but there's a question online. Shall I read it? Do you think any artists have been misled by the information-oriented, gene-centric view?
I think misleading is a very heavy term. I don't think. I think artists. I love art, and I'm sure for people who know me, I love talking to artists because I can learn from them. And artists, at least the ones I talk to, I respect them because they create things without a purpose in their mind. They're just driven by curiosity. And I think when you're curious, all information, right and wrong, are useful, equally useful. And I think this kind of, and that reflect in the artwork. And because there's no right and wrong in the artwork, and artwork is just a reflection of these artists or the society's, basically the time and the artist's mind. So I think in a way, art is a mirror, right, to the time. So, and therefore, there's no, I think misleading is kind of a strong word. I do think the people who know me knows that I have a very passionate feeling about the way we teach biology. I think we are not teaching them our children right. But there's another discussion, right? So,
any other questions?
We can talk about foreskin over dinner, but I put this slide there because you can email me by using this QR code. I love to collaborate with artists. So, in fact, we have artists in residence coming to working in our lab a week on Monday. So, if you'd like to discuss with me, or even for artists who want to come to our lab and basically have a conversation over coffee, or even doing some experiment together, you're welcome to contact me.
Perhaps you should have called your talk "Foreskins and Earlobes."
Yeah, that's.
You would have got a bigger audience, I think.
Yeah, so we have talking about model. Oh, we have to go.
Yeah, we have to wrap because we're on a tight online schedule here. All
right. Thank you, Rosa and Yun, and also previously David, who is with us. So this is a very interesting but short discussion. I'm sure we will continue to discuss on the scientific aspect and also angle on the topic of machine learning in art and also bioinformatics in the rest of the conference and after that. So thank you.